How's it going, everyone? Vincent here, and in this video we are going to be doing a deeper dive into antibodies. And antibodies are things that your adaptive immune system uses in order to identify antigens. And antigens can be pathogens like the coronavirus that's going around right now. And so antibodies are these proteins that are very specific to particular features that exist on the surface of bad guys. And if so, we were to take a coronavirus, for instance, we would see these spike proteins, and at the edge of these spike proteins, we would see these little caps on them, and these caps are called receptor binding domains. And so the receptor binding domain is the thing that is most exposed on the outside of the coronavirus. And so what an antibody would do, hopefully, is that as it's patrolling around in your body, it's going to see this particular feature, whatever features exist on the receptor binding domain of the coronavirus, and it's going to hopefully have affinity for it. And if it has affinity for it, that means that it's going to stick onto the outside of the coronavirus, and it's going to stay there. And it's going to have two effects. The first effect is going to be it's forming this physical barrier that will prevent the coronavirus from using its spike protein to actually infect any new host cells. So it's neutralizing this particular pathogen, the coronavirus. And in addition to that, it will make it very easy for dendritic cells or macrophages or other immune cells that you have to recognize that this is obviously a bad guy, because I see that whoever this guy is, he's got this antibody stuck on the outside of him. So this is obviously a bad guy. And what a dendritic cell would do is it would come by and it would basically eat it. It would perform this thing called phagocytosis, in which it recognizes this antibody and it would know that whatever it's stuck onto is clearly something that's not yourself. It's clearly something that it needs to get out of you and basically kill. And so the question here is, what makes antibodies different? And you have many types of antibodies. I only have 4 colors here, so I'm doing the best I can with what I have. But if we were to do a deeper dive on antibodies and zoom in on this stuff, what we would see, if we were to take this blue antibody, for instance, is we would see some similarities and some differences to other antibodies. Every antibody, and this is an IgG antibody specifically, has symmetry to it. And what this means is that basically, if this is a piece of paper, you could fold it and it would be a mirror image of itself. And within this antibody, we have something called a heavy chain, which is this blue thing, as well as a light chain, which is this red thing. And within the heavy chain and the light chain, there are constant and variable regions. And the thing that makes these two antibodies different from each other is their variable regions. And so your repertoire of antibodies that is roaming around inside of you right now is different from one another because of these variable regions. These variable regions are variable because of the VDJ recombination that occurs in the plasma or the B cells that are developing. And so because of this VDJ recombination, you get different types of amino acids present on the actual antigen binding site, which is another way of saying the variable heavy and variable light chain regions. And so with this, you get antibodies that basically have affinity for different things, because what your immune system has to do is recognize basically anything that isn't yourself. And so these antigen binding sites have to be able to recognize basically anything. And so it's a huge job. And so you need an enormous repertoire of antibodies roaming around. And I lost some of my antibodies, but basically you've got these antibodies that are roaming around, and hopefully one of them is going to have some kind of affinity for the coronavirus. These antibodies and the types of amino acids that go into those variable regions make them different because these amino acids have tons of different features. Like, they can either be positive or negative or acidic or basic or hydrophilic or hydrophobic. And so you've got these different types of amino acids that give these types of different characters. And so if you were to think of something like magnets, if we knew that the receptor binding domain on the coronavirus had this positive charge, if the variable region on our antibody had a negative charge, it's going to do a great job of basically just sticking on and latching onto the coronavirus really well, because there's going to be that electric attraction between the two. And so because the particular antibody, this red antibody, had that nice feature that gave it a very nice binding affinity, a strong attraction to a key feature that is easily accessible on the coronavirus, it's able to stick onto the coronavirus and block it from infecting any new cells, as well as make it very obvious to your immune system that this is a bad guy that you need to kill. Whereas another antibody in your body that's produced by another B cell might have a positive charge, and so because the coronavirus has this receptor binding domain that is, in this example, positively charged, they're just going to repel each other. So this antibody wouldn't really do that great of a job. And so what scientists and researchers are doing right now is we're basically trying to figure out what types of antibodies can we make that will have high affinity for specific proteins that are on the outside that are very accessible on this coronavirus. And once we can do that, we can start making a ton of these antibodies and give them basically as vaccines to people, so that once you have this antibody roaming around in your system, if you ever come into contact with the real coronavirus, it's going to do a great job of sticking onto it, labeling it, and helping you beat it. And so that's going to wrap things up for this video. I hope you guys find it interesting. Let me know if you have any questions. Please stay safe and wash your hands, and I'll talk to you next time.